Hi everyone. Today we are going to have lecture 7. This is the last lecture before our first exam. So today we'll discuss complex traits. When Mundell was working with garden peas, each pair of traits only had 2 phenotypes. For instance, seeds are smooth or wrinkled, and green or yellow. There was nothing in between. So for those phenotypes that fall into 2 or more distinct, non-overlapping categories, we name the phenotypes as discontinuous variation. In contrast to that, the phenotypes that are distributed on a continuous fashion, we name the phenotypes as continuous variation. An example is the height of a tobacco plant. It has not only tall or dwarf, but also has many intermediate heights. Mundell fortunately did not choose tobacco for his study. This is a graph comparison of a trait such as a height that shows discontinuous and continuous phenotypes in different plants. Shown in A, the histograms shows the percentage of plants with different heights in cross between tall and dwarf strains of the pea plant. The F1 generation you show here has the tall phenotype, but then they don't have the dwarf phenotype. And the F2 will have 2 distinct phenotypes, that 70% of the offspring are tall and 25% are dwarf. In the panel B, the histograms show the distribution of height in tobacco plants. Interesting in the F1 generation, all the plants are intermediate height. And in the F2 generation, it shows a range of phenotypes from dwarf to tall. So in tobacco plant, the height is a continuous phenotype. This picture is another example of a continuous variation. So this is a group of biology students organized according to height. Then the question is how a continuous phenotype is produced. The continuous phenotype is caused by complex traits. We have learned that genes can control traits by the central dogma, right? So the genes can produce protein at the end. So traits, phenotype, is an expression of proteins, a combination of different proteins. And all Mundell's laws were based on the assumption of one gene controls one pair of traits. This is the reason that in his experiments, we only saw discontinuous phenotypes. We call these types of traits as monogenic traits. However, in higher organisms, such as a human being, many traits are controlled by more than one gene, or even factors other than genes, okay? So we call this type of traits as complex traits. If the trait is controlled by more than one gene, we call it a polygenic traits. If the environment factors also contribute to the appearance of the trait, we call these multifactorial traits. So the previous picture shows the human height. That is an example of multifactorial traits. Polygenic inheritance usually follow a bell shape or called a normal curve. In a normal curve, few individuals are at the extremes of the phenotype, like here or there, and most individuals are clustered around the average value. The picture showing different heights is exactly distributed like the normal curve. There are several characteristics about complex traits. First, complex traits are usually quantified by measurement rather than counting. Mundell could count the pea plant because he could differentiate colors, heights of the plants easily, as all the phenotypes he studied were discontinuous. But complex traits are continuous and impossible to classify by just counting. Measurement is the more precise way to quantify. Okay. Second, to establish complex traits, at least 2 or more genes contribute to the phenotype, although the contribution from each gene may be small. Third, because phenotypes vary across a wide range, in order to study the traits, it would be best to study in populations rather than in individuals. Let's see more examples. Skin color is a complex polygenic trait controlled by 3 or 4 genes, producing a wide range of phenotypes. Environmental factors, such as you exposed to the sun and weather, also contribute to the phenotypic variation, making this a complex multifactorial trait. So remember, multifactorial traits are determined by multiple genes and environmental factors. For multifactorial traits, the environmental effects are essential factors. However, environmental factors do not change DNA sequences. Then how can they contribute to the phenotype? Again, phenotypes or traits are determined by proteins made in our cells, right? So then the different traits are determined by different proteins or the amount of proteins our cells make. This indicates that gene expression can be altered without changes on DNA sequence. This regulation is named as epigenetics. So environmental factors contribute to traits through epigenetic modifications. Later, scientists found that epigenetic modifications mainly occur on the histones or the DNA sequences. So we will have a designated lecture introducing epigenetics. I will not go into details today. There are 2 established ways to study the multifactorial traits. One way is to use the threshold model as shown here. In this model, all of the factors that influence the development of multifactorial disorder, whether this is a genetic or environmental, can be considered as a single entity known as genetic liability. In a general population, the distribution of liability is also a bell-shaped curve. The liability is caused by the number of genes, each acting additively. Only individuals above certain threshold shown here then affected by the disorder. So here you can consider the threshold as a threshold for expression of abnormal phenotype. In the general population shown here, the proportion beyond the threshold is the population incidence. And among the relatives, the proportion beyond the threshold is the familiar incidence. The curve for relatives affected will be shifted to the right. So the familiar incidence is higher than the general population incidence. Based on the liability curves, you can tell that the evidence for threshold effect can be revealed by comparing the frequency of the disorder among relatives in affected families versus the general population. This table shows the familiar risks for multifactorial threshold traits, such as like a clubfoot and a cleft lip shown here. So you can see that the risk for the disorder decrease as the degree of the relatedness decrease. Using clubfoot as an example, the monozygotic twins have 300-fold of higher risk than general population, and the first degree relatives have 25 folds. That's much less. while second-degree relatives have even lower risk, only 5-fold. And you can see the third-degree relatives only have 2-fold more of risk, which is quite close to the general population. These examples tell us if you marry to a person with very different genetics as you are, the chance of your child having genetic diseases is less. In the old days, in many countries, people like to marry to their relatives like cousins, which was the one major reason for rare familial diseases. Another way to study multifactorial traits is the recurrence risk. The recurrence risks are empiric risks derived from populational studies. Listed are several factors that contribute to recurrence risk determination. First, we talk about consanguinity. As related parents have a higher risk of having an abnormal child. Second, they previously affected the child. This is easy to understand. If the parents had affected a child before, then the risk for them to have another affected child is greatly increased. In addition, if the affected child has a severe phenotype, shown here is the severity of defect, so indicating the genotype is well over the threshold of genetic liability, then the risk is even higher for having another affected child. Lastly, the higher frequency in one sex. That means the recurrence risk varies on different sex. For instance, a lot more males have color blindness than females, indicating the threshold must be higher for girls than boys. Now we learn that multifactorial traits are contributed by both genes and environmental factors. Then an interesting question is, for one certain disease, how much do genetic factors or environmental factors contribute to the trait or phenotype? Either a way to quantify it. To do this, scientists can measure the phenotypic variation. Phenotypic variation is the variability in phenotypes that exists in a population. For example, people come in all shapes and sizes, like height, weight, and body shape. Those are phenotypes that vary, right? Measuring phenotypic variation in populations rather in individual height highlights the interaction. Another way to study multifactorial traits is the recurrence risk. The recurrence risks are empiric risks derived from populational studies. Listed are several factors that contribute to recurrence risk determination. First, we talk about consanguinity, as related parents have a higher risk of having an abnormal child. Second, previous affected child. This is easy to understand. If the parents had an affected child before, then the risk for them to have another affected child is greatly increased. In addition, if the affected child has a severe phenotype, indicating the genotype is well over the threshold of genetic liability, then the risk is even higher for having another affected child. Lastly, if the condition is more common in individuals of one particular sex, recurrence risk varies on different sex. For instance, A lot more males have color blindness than females, indicating the threshold must be higher for girls than boys. Now we learned that multifactorial traits are contributed by both genes and environmental factors. Then an interesting question is, for one certain disease, how much do genetic factors or environmental factors contribute to the trait or the phenotype? Is there a way to quantify it? To do this, scientists have measured phenotypic variation. Phenotypic variation is the variability in phenotypes that exist in a population. For example, people come in all shapes and sizes. Height, weight, and body shapes are phenotypes that vary. Measuring phenotypic variation in populations rather than in individuals highlights the interaction between genetics and environmental influences on phenotypes. The phenotypic variation is contributed by genetic variants and environmental variants. Then, how should we measure this? We can use two values to determine the influence of genetic and environmental factors on traits. The first is heritability. Heritability is a measure of how differences in people's genes account for differences in their traits. If heritability is 1.0, meaning genetic factors contribute 100% to phenotypic variation. But if the heritability is 0, meaning no genetic contribution to phenotypic variation. Instead, environmental factors contribute 100%. Okay. Another measurement is correlation coefficients. In statistics, a correlation coefficient is a numerical measurement of some type of correlation, meaning a statistical relationship between two variables. In genetics, it can be used to calculate heritability by showing the degree of genetic relatedness between two individuals. For identical twins who share the identical DNA, the correlation coefficient is 1. Whereas for children and parents, since children receive half of DNA from each parent, the correlation coefficient is 0.5. One of the most valuable tools to understand multifactorial traits are twin studies, particularly identical twins, because twin studies can separate and evaluate the genetic and environmental factors responsible for phenotypic variation. As identical twins have the same genetics, if they are separated at birth and grow up in different environments, this case can be used to analyze how environmentals contribute to any traits observed in the identical twin. Vice versa, if adopted children living in the same family, the case can be used to evaluate how genotypes affect the trait, as the children have the same environment but the different genotypes. However, there are less frequent chances that twins result from the independent fertilization of 2 eggs by 2 sperm at the same time. Usually, one ovulation releases 1 single egg, but somehow 2 eggs are released at the same time and both get fertilized. Although these 2 embryos simultaneously occupy the same uterine environment, they share only about half of their genes. We call these twins as dizygotic twins. Dizygotic twins can have different genders and do not look like each other that much. Therefore, if the twins grow up in the same family, we can consider that monozygotic twins share 100% of their genes and the same environment. And the dizygotic twins share half of their genes and the same environment. Since the environment is the same, The greater the heritability of a trait, the greater the phenotypic similarity can be observed between monozygotic twins than dizygotic twins. The height is an example. If height is mainly determined by genes, then the identical twins will have more similar height than dizygotic twins. But if genetics does not contribute much to height, then the height variability will be the same in identical twins, that's monozygotic twins, and in dizygotic twins. Related to that, I need to introduce a new definition to you. That is concordance. In genetics, concordance means the presence of the same trait in both members of a pair of twins. But if only one member of a twin pair has the trait, we say the trait is discordant. The greater the difference in concordance rate between monozygotic and dizygotic twins for a given trait, the greater the heritability. We'll see several specific examples next. This table shows concordance values in monozygotic and dizygotic twins. For a trait controlled by completely by genetics, the concordance rate should be 1.0 for monozygotic twins and 0.5 for dizygotic twins. From this table, you can tell that blood types, eye color, and mental retardation, those are highly concordant in twins, meaning genetics largely contribute to these traits. In contrast, the concordance value of cleft lip are quite low in both monozygotic and dizygotic twins, indicating environmental factors largely contribute to this phenotype. Now let's discuss how we use these tools to study specific traits. First, let's see if obesity is a complex trait and whether environment contributes to obesity. I guess you already have an answer in your mind. This table combines several obesity studies in twins. Here, heritability is calculated. We learn that the higher the heritability, the less contributed by environment. When heritability equals 1, the trait is 100% controlled by genetic factors. When heritability equals 0, the trait is 100% controlled by environment. You can see that in different studies, the heritability is between 0.6 to 0.9, indicating that obesity is affected by both genetic factors and environmental factors, and genetic factors weigh more. One useful tool to study obesity is mouse model. Scientists found a gene named OB, which encodes a weight control hormone named leptin in mice. Leptin is mainly made by adipocytes, that means fat cell, and enterocytes in the small intestine that helps to regulate the energy balance by inhibiting hunger. So leptin has another name as energy expenditure hormone because the action of leptin needs the binding between leptin and its receptor. When scientists disrupt either leptin gene, that's the OB gene, or leptin receptor gene, that's the DB gene, the leptin signaling pathway will be disrupted. Therefore, these mice will show obese phenotype. Showing in this picture is the OB mouse on the left. Its OB gene is mutated and no leptin is made. On the right here is the normal mouse as a control. You can see indeed the OB mouse is quite obese. Scientists hope that this study could provide clues about how weight is controlled in humans. This slide shows how leptin is produced in fat cells, or we call it adipocytes. move through the blood and binds to the receptors in the hypothalamus. If one person has a lot of fat cells, the cells secrete leptin, which controls the weight by regulating the conversion of food or energy into fat and the rate of energy consumption. Since the identification of leptin and leptin receptor, people got very excited and thought we might be able to solve the obesity problem once for all. But things were not always as easy as we thought. Similar to mice, human beings also have the leptin and leptin receptor genes, and they were named as LP for leptin genes and LEPR for leptin receptor genes. When genetic sequencing was done on those obese individuals, scientists found that mutations in all the genes involved in the leptin pathways only account for approximately 5% of obese cases, strongly indicating that obesity is a multifactorial trait. Changing leptin pathway might reduce obesity in maximum of 5% of affected individuals. So apparently it is not an efficient way. With the development of genomic sequencing technology, more genes related to obesity have been identified. This slide shows some obesity-related genes that have been identified. The green triangles shown here, those represent single genes in which obesity is the main phenotype with little environmental influence. The red triangles shown here, there's actually more, represent genes that are candidates for obese genes. Mutations in these genes have an association with obesity. The red bars, there are several of them. They present chromosomal regions that may contain other genes associated with obesity. More than 70 genes associated with obesity have been identified. We learned SNP before, and we know that in order to identify SNP, we need to sequence the DNA, right? So many SNP were identified through a study called the GWAS, okay? So that's genome-wide associated studies. GWAS is an approach used in genetics research to associate the specific variations with particular diseases. The method involves sequencing the genomes from many different people and looking for genetic markers that can be used to predict the presence of a disease. Many obese-related mutations we just mentioned, we just mentioned were identified through GWAS. For this type of studies, you may recruit two groups of participants. One group may be obese people, and the other group may be normal people as the controls. Each person gives a sample of DNA from which millions of genetic variants are read using SNP arrays. If one type of the variant is more frequent in people with obesity, the variant is said to be associated with obesity. The associated SNPs are then considered to mark a region of the human genome that may influence the risk of the disease, all right? So SNPs do not always exist as a single nucleotide variation, okay? Sometimes several SNPs are inherited together as a combination, and we call them as haplotype. So let's see how haplotype looks like. So this picture shows how haplotypes look like. See here, we have 4 SNP. They are organized into blocks that are always inherited together. So you just remember the haplotype, what does it mean? Obesity has been a problem for developed countries such as the United States. So this picture in our textbook show a fraction of obese individuals by states. This is in 2012. No states have obesity rates below 20%. See here. And only 9 states have rates between 20 to 24%. Texas was not the worst state, as the percentage of obese people fell in the category of this 25% to 29%. There are some states that more than 30%. But then this is updated image I found in the CDC website. This is 6 years later, 2018. Do you think the situation getting any better? Now the Texas used to have 25 to 29%, right? But now it increased to 30 to 35%. It's not getting any better, at least for Texas. So the most striking statistics came from the percentage of obese children here. In 2016, Texas was one of the 2 states that had the highest percentage of obese children aged this from 10 to 17. The variability of obese population in different states also indicate that environmental factors contribute to obese phenotype. For instance, in Texas, we know that the food is more affordable, and then maybe the hot weather and the mosquitoes restricted many outdoor activities. And in the near future, we will probably see a higher percentage of obesity in the countrywide due to COVID-19. So these are all good examples of how environmental factors affect the traits. All right, so we discussed the human height at the beginning of this lecture, and know that height is a complex trait. It has a heritability of about 80 to 90%, indicating that height is largely controlled by genetic factors. So far, over 200 genetic variants have been identified that contribute to growth and adult height. The ways to identify these genes, or SNPs, include the GWAS we just learned, or looking into mutations in individuals with abnormal heights, either very short or very tall. Diseases with skeletal defects could give out some clues as well. If new genes or new signaling pathways can be identified, scientists might be able to design therapeutic intervention to treat growth disorders. Can you predict any genes that are involved? We know that the height is mainly determined by the growth of bones. Therefore, any genes that affects bone growth is likely determining the human height, right? For instance, any gene encoding growth hormone receptors, transcription factors that drive the growth of bone cells are probably all related. Similarly, skin color is also a complex trait, which is contributed by both genetic factors and environmental factors. Skin color variation is one of the most striking examples of human phenotypic diversity. It is dominated by melanin, a pigmentation located in the base of the epidermis and produced by melanocytes. Melanin has 2 major forms. One is the phenomelanin. It's kind of yellow and reddish color. And the other is eumelanin, which is a black and brown color. Phenomelanin is mainly produced in the light complex people, whereas eumelanin is most produced in the dark complex people. In addition, the number and size of melanin particles differ among individuals, and is probably even more important than the proportions of the 2 forms of melanin in the determination of human skin color. All right, so in addition to genetic contributions, environment also contributes to skin color. It has been found that skin color varies with latitude, not longitude. Let's see water map next. In this map, you can clearly see that human skin color is darkest near the equator and is progressively lighter at higher latitudes. Fundamentally, skin color is correlated with the distribution of UV radiation. Populations closer to the equator tend to have a darker skin for protection against the UV, since overexposure to UV may decrease folic acid levels and cause skin cancer. In contrast, the light skin in populations at higher latitudes is under selection to maintain vitamin D photosynthesis, which is UV-dependent process. Then how about intelligence? Is intelligence an inheritable trait? Historically, it was believed that physical dimensions of regions of the brain were a measure of intelligence. This was a model showing areas of head overlaying brain regions that control different traits. Intelligence was estimated by measuring the area of the skull overlaying the region of the brain to control this trait. So the bigger your head is, the smarter you are. However, now we know this is not the case. As a matter of fact, human intelligence is also a complex trait, meaning it is contributed by multiple genes and environment. The inheritability of IQ is between 0.6 to 0.8, suggesting that genetic component has a significant impact, but not all. Also, when heritability was determined in twin studies, heritability decreases in monozygotic twins when they raised in different families, confirming that environmental factors also contribute to intelligence. This slide shows a graphical representation of correlations in intelligence measurement in twin studies. The purpose of this study is to see if intelligence is controlled by genetic factors or environmental factors. What is the expected value shown here? Yes, the expected correlation coefficients are purely determined by genetic relatedness in each set of individuals. The bars shows here the range of correlation coefficients, and the vertical lines showing here are the mean, like median value. So the numbers in the parentheses over here are the numbers of twins pairs they studied. I wish this figure could also indicate the statistics of significance, which is required for publications these days. This diagram gives out the two pieces of information. The first is that intelligence is genetically inheritable because the expected value and the measured value are pretty close, okay? Second is that environmental components contribute to heritability. For example, if you compare these two bars, the last two bars, the monozygotic twins raised together, this is have a higher correlation than the monozygotic twins reared apart, indicating environmental factors contribute to the difference. Therefore, the human intelligence is a complex trait. Now let's see several questions together. Okay, the first one, one goal of a pedigree analysis is to determine what? So you can pause here and take a look at it to get the right answer. Okay, the correct answer of this question is D. Let's see one by one, okay? We have gone through a lot of pedigree analysis, and it did not give out detailed information on what exactly the disease is, or chromosome localization of a gene, or provide any option on disease treatment, shown in the last option. But it does show the inherited pattern as such dominant or recessive. So D is correct. Let's see the second one. If a pedigree of several generations shows only females affected by a particular trait, it cannot be a what trait? So you can stop here and then, pause here and think. Okay, so this question has two keywords. One is females, another one is cannot. Okay, females do not have Y chromosome, therefore there's no Y-linked trait. So the correct answer is C. Okay, see this one. So the X and the Y chromosomes, what? You can pause here again. Okay, so the X and Y chromosomes are sex chromosomes, right? So A and B are related to autosomes. Actually, A and C are related to autosomes. Therefore, they are wrong. So, but in B, color blindness is X-linked, right? So, but its inheritance pattern is recessive. If it's recessive, then the carriers do not show disease phenotype. So B is not correct. So only left one correct answer is D. Okay, the second one. Mitochondria are transmitted from mothers to, okay, you can pause here. So this is quite apparent that C is the correct answer because mitochondria and the mitochondrial DNA are passed from mothers to children from cytoplasm of the egg. We learned about that. Mitochondria in the cytoplasm, therefore their DNA pass is also from cytoplasm. Okay. Okay, the third one. When affected males produce all affected daughters and no affected sons, the pattern of inheritance is likely to be what? So this is a fill in blank question. So you can pause here again, and then we'll talk about it. Okay, first, the daughters and sons are different, right? In this affected daughter, but no affected sons, indicating this is sex chromosome related disease, right? Because son is normal, no affected sons, then the son's phenotype will be, genotype is going to be X and Y, right? But then here it says the father is affected. So meaning his X chromosome has mutation because he contributes a Y to his son. So the father has to be X and Y. Then here, all daughters are affected, indicating the daughter is X and X. Heterozygous daughters are affected, indicating this is a dominant trait. So the correct answer is all affected. So the correct answer is what? So this is a X-linked dominant inheritance. But then here you can, some people probably will argue, then the daughter could be XM and XM, right? It's all affected. But then if that's the case, the answer will be X-linked recessive inheritance. Would this happen? No, because under that situation, the mother has at least to be XMX. Then they will produce affected son because the mother contribute X to their son, right? But here, all sons are not affected. So then the only correct answer is X-linked dominant inheritance in this question. All right, so I hope you got every question correct. Okay, now we will talk about exam 1 this coming next Monday. Okay, the time is next Monday, 4 to 5:30. It's an open book exam. And by the time before the actually a day or the same day, earlier the same day, I'm going to provide a link in the announcement by 4 p.m. you will be able to see those, okay? So this exam weighs 20% of your final grade, and you are going to do 50 multiple choice questions. You have 40 minutes to do it, okay? The exams, you can start at any time between 4 to 4:50 because they're going to be, the exam will be auto-submitted after 40 minutes. If you don't submit yourself, they're going to be auto-submitted. Questions are based on first 7 lectures we already have, and the questions will be randomized, okay? So everybody will get a different order of exam. The lecture slides and videos are both important. Don't just look into the slides, but if you watch the video, you know that we have some YouTube videos inserted into our lectures, and they are important too. All right. So scores will be given right, not actually, the scores you will see it right after your exam. Okay. So you will see how much you get. This is based on 100 of scale, okay? And then correct answers will be provided at 9 p.m. I mean, if you want to know why you get this wrong, and you can look at it later in the night. And then at the same time, I will schedule a Zoom meeting. I will provide a Zoom meeting link together with the link with the exams. But you don't have to go to a Zoom meeting unless you have questions, because otherwise you can focus your questions, okay? Then lastly, you want to make sure that you have a good internet connection, because that's the reason I provide also practicing test, sample test. You can practice to get familiar with all the styles. But if your internet has a disruption during your taking the exam, that's not a good thing, because these 40 minutes include troubleshooting the technical issues, okay? So you don't want that to happen. Find a good site that has a good internet connection. So don't argue with me about the internet connection later on. I'm not going to accept that, okay? So this picture is before, when we didn't have pandemic. Everyone can have a test in the classrooms, and then they can focus on their own questions. However, this semester we moved to the classes online, and you could say, oh, yay, let's take tests online as a group, like that, okay? I want to emphasize that group testing is absolutely not allowed, okay? No group testing. You got to focus on your own test. All the questions will be randomized, so you could waste your time discussing any questions, okay? Since you already spent money on taking this course, taking this opportunity to learn it really well, you will benefit from this learning, I guarantee you, okay? So that's all for today. I wish everybody can have a really nice test. I'll see how it goes. All right.